Hello, everyone. So today we have Jerome Roehm speaking on an introduction to topological data analysis and hippocampal place cells.
Thank you very much. Good morning or afternoon to all of you. Thank you so much, Sarah, and the organizers for having me. I'm looking forward to this talk. As I said in the abstract, it's kind of a 2-for-1 special, with the first part focused on an introduction to TDA. We'll cover the mathematical foundations. The motivation for this is in part selfish for me, because this is developing into a talk that I'm hopefully going to give as a general audience. What do you want to do in this department when I apply for jobs? And so it needs to be general enough for the science faculty as a whole to understand it, but also interesting enough for those interested in topology to follow along. This is a kind of more central data science portion of it. I know lots of people come from more pure or abstract topology backgrounds, but this will hopefully be very concrete and have lots of pretty pictures. The second part of my talk will center on my research into hippocampal replay and preplay in place cells in the hippocampus of mice and rats. This picture that you see here depicts a rat running along a track with the position on this axis, and then we have the firing rate of several neurons plotted here. And you can kind of see, certainly here with neuron 43, it's much more active when the rat is between 350 and 400 on the track. The discovery of these things is basically what won the Nobel Prize in 1971 with John O'Keefe. And so I was just a few generations too late to collect on the prize, but I can stand on their shoulders and do more research. All right, so introduction to TDA. Here we seek to understand the structure of data through topology. The data that we sample may come from a circle, a sphere, a torus, Klein bottle, any of our favorite topological shapes. And we seek to better understand the mechanism behind the data. Why does it form a circle? Why does it form a sphere? How can we detect that in higher dimensions when we can't plot it or visualize it in 2 dimensions or 3 dimensions? Then we can also feed this into perhaps neural network or other things to try and differentiate between types of data. Is this data collected from a sick patient or a healthy patient? Is this data collected from male or female organism? And things like that we can help use to differentiate types of data. So the motivating example that I'll run throughout this talk is one of a muntjac deer. This is a completely fabricated data set that I've done very carefully to make a few key points, but it'll give your imagination something to run on. So suppose we have the GPS coordinates in 2 dimensions here of a muntjac deer over several days. They're kind of an Asian miniature deer. This is a former student of mine that worked at a zoo that sent me this picture, part of the motivation. Very cute little animals. So we have the GPS coordinates. Here in 2 dimensions, we can easily visualize it, and we can easily pick out kind of the 2 major holes, the 2 major cycles, right? And we want to develop a way of numerically finding that with topology. Our desire is to identify the 2 major holes that I just pointed out, and this will hopefully give us some insight, or the conservationists or naturalists some insight into maybe the den location or what obstacles, maybe they're going around a predator seeking food or water. And it can also differentiate between maybe healthy or sick, male or female, what their behavior is like in mating season versus other seasons based on the analysis of the data that we collected. Again, this data in 2 dimensions, so we can easily visualize it. However, methods hold perfectly well for higher dimensional data. So this is going to be a high-level overview of the TDA process. First, we're going to, in trying to identify these 2 circles, we're going to do what our mind's eye does and just connect the dots, right? Just like we were 6-year-olds. But in order to connect the dots, we have to say, connect the nearby points. What is nearby? What metric are we using? What metric space are we in? Does it live naturally in Euclidean space, or is there some kind of other metric space where the data lives? Do we want to use spherical radii to develop, or do we want to use maybe the 1 norm or the infinity norm to try and measure how close our data points are? Is one axis stretched or compressed? We may have to take that into account with our data. And then once we define that, we have to choose, well, what choice of radius? What kind of radius do we want to connect? If we choose this radius, we capture this hole, as we can see. But if we choose a larger radius, we capture the big hole and the small hole fills in. So we have to answer the question, what kind of choice of radius are we going to connect the points? And keeping in mind, we have to have this to be computable. I know people that work with hundreds of thousands of points in data clouds, and it doesn't do us any good if our methods aren't able to run in a reasonable amount of time. So after we decide all of those things, we're going to study the space forms. And of course, as a topologist, I am legally obligated to inform you in all of my talks that a donut is the same as a coffee cup. Right? They have the same topological features, the invariants, the holes. We're all familiar with those in this space, so I won't dive deep into them. Just fulfill my legal obligation. So the zero-dimensional holes, zero-dimensional homology details connected components. On this slide, there's 13 of them that you could count up if you were a fast counter. One-dimensional holes or one-dimensional homology indicates an enclosed disk, like in the middle of a circle. Two-dimensional homology indicates an enclosed void, like the inside of a sphere, right? And we could keep counting on up if we so desired. So this is going to be the simple summary. We'll go back and update this here in a second. So we assume our data comes from some sort of topological structure, some kind of manifolds in some kind of metric space. And then through our sampling, we develop a point cloud of the data. Once we're given the point cloud, we connect the points in the way I just mentioned. We choose a radius, we choose a metric, and that develops into a simplicial complex, which I'll give you the details about here in just a second. Once we have that, we can do some linear algebra to compute homology, to compute those holes. And then after that, we can either do some analysis and try and uncover the total topological structure that the data was collected from, or we can use it to predict and classify what kind of data we have. So let me tell you about the details of a simplicial complex. So simplicial complexes are defined, stored, and computed as combinatorial objects, just sets and subsets, nothing more. I'm going to be showing you the geometric realization of these to help you understand what I mean by holes, and it's easy to go back and forth between the geometric realization and the combinatorial definition once we see these. So a simplicial complex is given a set of vertices, it's a collection of subsets, right? So given all the vertices, we're going to have a collection of subsets, and we're going to play by two rules. Those rules are, for each vertex, we have to have the singleton, and we have to obey downward closure. The subsets also have to belong to our collection, in other words. The elements are called simplices, and if we have sigma as a subset of tau, we say sigma is a face. The dimension is the number of vertices minus 1. That'll be clear when I show this picture. So these are simplices or simplexes. A zero-dimensional simplex is just a point. One-dimensional simplex is a subset of 2 points. You can think of that as a line segment. Two-dimensional simplex is 3 points all connected, so a solid triangle is the geometric realization of that. Three-dimension is 4-point tetrahedra filled in solid. Right, so that's how we define simplicial complexes and go back and forth between the geometric definition or geometric realization and combinatorial definition. So we'll have this miniature example that will run here showing how different choices of radii will give us different simplicial complexes on this point cloud. So if we have no radius, we just have the zero simplices, just the points. If we decide to connect it via small radius, we add in some one simplexes, some line segments. Medium radius, now we have a 2 simplex here, the solid triangle, and we've also added some additional 1 simplices. If we go up to a larger radius, then we have this solid tetrahedra here formed by this 3 simplex, along with a solid triangle glued onto it, which is a 2 simplex. And again, notice we're obeying all the downward closure requirements that we have from this 3 simplex. Right, so that's how we define and compute with simplicial complexes. Now let's try and decide how we're going to connect points to go from our point cloud to a simplicial complex. There's 2 major methods that we do with these, 2 methods that I'll talk to you about today at least. So given a point cloud and a radius, a certain subset will form a case dimensional simplex, i.e. we're going to connect those points if we use the Vietoris-Rips method, if the distance is less than or equal to 2r for all of the points. So notice between B, D, and C, pairwise, the distance is less than or equal to 2r. The circles overlap. So we have this 2 simplex made of these 3 points. Pairwise, the distance is less than r, so we fill it in. Right? It's a complete clique, in other words, in this graph. We fill that in. In the check complex, we connect the points if there exists some common third point or fourth point or other point, such that the distance to that point to all of the points of interest is less than or equal to r. So in this case, the triangle is not filled in because there is no overlap in the Venn diagram of these 3 circles. And so we do capture another cycle here, another hole in the check complex. You can easily observe the check is a subcomplex of the Vietoris-Rips complex. That's an easy observation to make. And there's benefits and drawbacks for each type. The Vietoris-Rips complex is easy to compute because we don't have to have any knowledge of the underlying metric space. It's completely defined by the pairwise distances. It's also efficient to store because we can essentially store it as a graph and then fill in all of the cliques, all of the complete subgraphs. And it's also the secret to a long life. Leopold Vietoris, who lends his name to the complex, died at a ripe old age of 110 years old. So you can use that as a mathematical fun fact to impress all of your friends at parties, if you go to the right kind of parties. Anyway, so the check complex, it could be more faithful in smaller clouds, because here maybe we do want to capture that this is a cycle, that we do have a hole here, but that's blind to the Viator's-Rips complex. It is harder to compute and store because we have to find that mysterious other point that's less than or equal to the radius for all the different points. So it's harder to compute and store. So most of the time, and for the duration of this talk, we'll be talking about the Viator's-Rips complex. Right, so that's how we're going to connect our points. We'll define our radius. We'll use the Viator's-Rips complex to define our simplicial complex. Now let's go through some basic linear algebra and how we define homology. So this will be our miniature running example for the talk. This is the data of the point cloud, those 5 points. The radius I have fixed at 1.45. And this is the entire definition of the simplicial complex. We have 2 simplex, a bunch of 1 simplices, and of course all of our 0 simplices, those points. So this is the data of the simplicial complex seen here in its geometric realization. And if we're talking about linear algebra, we of course need a field. And we'll use the simplest field that we know, the field with 2 elements. Nice to compute and store with. So a few definitions. For any dimension k, the kth chain group is a free vector space over F, with each k simplex as a basis element. So the second chain group is a free vector space with just our single 3 simplex in there. Right, this B, C, D. Right, so it has all linear combinations of these. Similarly, this 1 simplex or 1 first chain group, all linear combinations of these elements over our field. All linear combinations of these form our 0th chain group. A few examples of chains. We only have a single non-trivial 2 chain, that's BCD. Here's a perfectly good 1 chain. It's a linear sum of our basis elements. Here's another perfectly good 1 chain. It's a linear sum of our basis elements. Somehow we need to detect the difference between this 1 chain and this 1 chain because this is a cycle, whereas the first one is not. So we'll talk about that here in a second. To do that, we'll define a boundary operator, which goes down the chain groups, which just picks out a single element. And why it's called the boundary operator and the intuition will become very clear here in just a second. So if we start with our 1-2 chain, BCD, we take its boundary and we see that 1 chain, right? Now you see why it's certainly called the boundary element. Algebraically, this is formed by just removing each of the elements in turn and taking the sum. Similarly, if we take the boundary of this 1 chain, after some cancellation, because we're in the field of 2 elements, we see its boundary is those 4 points. Here we have our other 1 chain that we would like to detect as a cycle, and we can see that after we cancel over the field of 2 elements, it has no boundary. And so now we have a way to define what a cycle is. Elements in the kernel, elements that map to 0, are k-dimensional cycles. For example, that's a 1-dimensional cycle, as is that. There's no 2-dimensional cycles because we only had a single non-trivial element. And all of the points map to 0 because they have no boundary, and so they're all in the kernel. Elements in the image of the boundary map are sensibly called boundaries. So this is a boundary because it's the boundary of that 2 simplex. Similarly, A plus C is the boundary because it's the boundary of that element of the first chain group. So we collect all these chain groups and these boundary maps and we form what's called a chain complex. And by a straightforward computation, you can verify easily with linear algebra. The image is contained in the kernel, and so we can define sensibly this quotient space, which we call the homology group. So homology, the key phrase is it cycles modulo boundaries. We're going to pick out all the cycles and we're going to quotient by the boundaries, meaning they're essentially 0. All right, so let's do an example with our mini example. So we're trying to compute the first homology group. The first homology group I told you at the beginning of the talk details 1-dimensional holes, i.e. the inside of circles. And so we'd like to detect somehow that we have this element in our 1-dimensional homology group. So using our mini example, I've shown all of the chain groups and the maps. The maps are linear operators, so we can express them as matrices once we fix the bases. And that's what I've done here. And we desire, again, to compute this H1. Kernel boundary 1, mod image boundary 2. So we can compute that the kernel of the first boundary map is shown here. Right, these are the two cycles. This is indeed a cycle. It's also a boundary. And this is also a cycle. Notice that when we compute the kernel, it's two-dimensional, but we could have chosen a different basis, as we know from linear algebra. Right? So we could also have chosen that orange instead of the red. In fact, we could have chosen any two of the three that I've shown, blue, red, or orange. We go to compute the image of the second boundary map, and that's pretty obvious, right? We had our 1, 2 chain, the boundary is BCD. In fact, well, the sum of those edges. So then when we take kernel mod image, as we've shown, the blue and the red mod the blue, we can mod by the blue, and that'll give us a class here that we see. So we have the red class, or if we happen to choose the blue and the orange as our basis, we have the orange class. So these must be the same by definition, and I'll show you why they're the same both geometrically and algebraically. So here we have the first homology group written in two different ways. The red class we could see as dragging across this blue 2-simplex to turn into the orange class. And that's what these blue brackets are, and that's why all of this geometry that I'm showing makes sense algebraically. Algebraically, we can look at it. This red class plus the blue element, once we cancel with the 2 in our field of 2 elements, we end up with our orange element. So both algebraic and geometrically, we have one element in our one-dimensional homology, which can be represented by either going all the way around or going around as shown. We can define the Kth-Betty number to be the number of holes, the dimension of HK. So in this case, we have one connected component, V0, one one-dimensional hole here, and no voids captured, no Betty 2. So we know how to sample our data. We have a point cloud. We can connect the points to form a simplicial complex using the Vietoris-Rips method. Connecting the points, though, we have to fix the radius. To fix the radius to form a simplicial complex, then we do linear algebra and homology, and then we can do our analysis. But how do we choose a value for R? Going back to our Muntjac example, talking about wanting to capture these two major holes, I'm going to go through some choices for R and some comments on them. So for a very small radius, we have 30 connected components and no holes. For R is 0.45, we kind of get a sneaky tiny hole forming here. We continue to increase the radius. Now we're down to 6 connected components. We have one one-dimensional hole captured. As we continue to increase, that hole fills in, and we have a single connected component with no holes. Increase the radius some more, we get a large hole formed, and it persists a while until we fill it all the way in. All right, so how do we choose a value for R? Kind of a trick question. We choose all of the values. All of the finitely many interesting values that results in homology changes, that is. And so we can update our simple summary to be a full summary. From our point cloud, we can vary the radius to form a bunch of simplicial complexes, plural. We do some linear algebra to compute what's called persistent homology. And then we can do more analysis to uncover the topological structure or predict and classify our data. So as we increase our radius, the connected components die, right? As they are connected, they die. And the holes are born and die. This information is encoded in a persistence barcode. So we have a certain fixed radius here that I've shown. We're capturing 2 one-dimensional holes, i.e. this one and this one. And we have however many lines we intersect in red, that many connected components. We'll go into this more later, but I do want to say that we've encoded this information. This is via one of the couple main theorems of TDA called the structure theorem. The details of the proof I won't go into now, but the main idea is it's going to give our direct sum of vector spaces the structure of an FT module. This M is the number of interesting radii that we have. And the T of V, this polynomial here, is a shift from one dimension to the next, describing how, from one radius to the next, describing how homology groups progress as the radius increases. This proof does invoke the decomposition of finitely generated modules over a PID, because F is a field, polynomials in one variable are PIDs, so we can invoke that algebraic theorem. However, as the field is trying to figure out right now, and when I say field, I mean lots of research going on right now, trying to figure out how this applies to multi-persistence. If we want to persist in 2 or more parameters, all of a sudden we cannot invoke the finitely generated modules theorem because polynomials in 2 variables are not PIDs. Right, so a little bit of algebra for those that are familiar with that, but just wanted to detail that this is a theorem. Persistence barcode only plays out so nice because of the structure theorem, and it does not apply to multi-persistence in the nicest way. So this persistence barcode, let's take a look at a few of the key radii here. So here the radius is small and we have a bunch of connected components surviving. As we continue to increase, we have our tiny hole born and die eventually. Then we have our small hole born and die. There is a radius where we have a single connected component and no holes. And eventually we'll close them all up again and close that hole. Right, so this barcode can also be expressed as a persistence diagram. Some people like one over the other, but it's the same data. We'll see a persistence diagram here in a second. So these barcodes are the main tool, or the main analysis that we perform, whether we plug those into a neural network or just try and deconstruct it ourselves. In H0, the average persistence in H0 can measure spread, diversity, or clustering. If the points are all together, the connected components are going to die pretty soon, whereas if they're spread out, it'll take longer. I did a video of this on YouTube, and if you're interested in basketball, I recommend it. My thesis for this was that the diversity as measured in playing styles has decreased as time has gone on in the NBA. So here we see the 1986 Celtics, the points are nice and spread out, whereas the 2016 Golden State Warriors, the points are more clumped. So the Celtics are more diverse as measured by average H0 than the Golden State Warriors. Going into higher dimensions, the length of the bars can be interpreted. Shorter bars are often viewed as noise, but not always. And longer bars are often interpreted as more significant. Right, so this very short bar here corresponds to this very tiny hole. Maybe this is just sampling noise. Maybe the muntjac was right here, but we didn't get a sample of it. If we had another point right here, that would kill that hole right away and it never would have formed. So there can be some sense to viewing the longer bars as having more importance, but not always. For example, here, this small hole may be just as interesting to us than the larger one, which has a longer lifespan. So a survey of some applications, I'll go into each of these briefly. I've already talked about the diversity in playing styles of basketball. Next, we'll talk about some high contrast patches in natural images, some disease diagnosis, biological pattern recognition, and some hurricane analysis. So just a quick overview of these. So Gunnar Carlson and a few of his friends sampled and filtered some high contrast patches from 5,000 images. So 3x3 patches, so we're living in 9-dimensional space. And after we analyze these 3x3 patches from the images and compile it together, the data appears to have been collected from around a Klein bottle, which is completely unexpected to start with. Really, really beautiful, but it kind of makes sense if we look at the diagram. So here we have 2 depictions of Klein bottle. We have a couple of different cycles on the Klein bottle that I pointed out. Let's start with the long blue cycle, the one that goes all the way around. Right, and if we trace the blue cycle from top left, progressing, and then where it's identified down here, and continue progressing, you can kind of see that that's the blue, gray, and white rotating around the center, right? It's rotating, if we trace it this way, it would be rotating in a counterclockwise direction. Right, so the main cycle there. Now, if you look at the red cycle, this has the same idea, but this is the black, gray, and white kind of rotating in a cyclic fashion as shown. Now the green, it's a little bit harder to see, but it's that same cyclic fashion, but turned vertically. And as we can observe from the Klein bottle, if we drag this red circle along the blue line, the blue axis, we end up at the green. Exactly. If we drag horizontal rotation along the rotation, around a rotation like that, we have vertical.
Like, is there any reason for the symmetry along in the images? Like from like the bottom left square is the same as the top right square?
Yeah, so that's the identification that forms the Klein bottle. So this edge here in black, you could think of it as sewing along this edge. So then we have a cylinder. And then we give that cylinder a little twist and identify these edges in opposite directions, which is what's responsible for the Klein bottle shape.
Did the diagram on the left come from the Klein bottle, or did this end up turning into the Klein bottle from some other image?
So this was after their analysis. They kind of pieced it all together.
Oh, cool.
So they computed the homology and said, oh, it has the homology of a Klein bottle. Can we sensibly make something that looks like it would come from a Klein bottle? And then they pieced this together in the paper and said, yes, all of the high contrast 3x3 patches are very close to some of these patches. And if you noticed, when we fold it up and make a Klein bottle, we have that characteristic. You can drag one cycle along one axis until you reach the other, and it forms the Klein bottle shape from the natural images.
Cool.
Yeah, super interesting and kind of beautiful, I think. So another application, this is one of the diagnosis side of things, is a group used some TDA to diagnose blockages in blood vessels. So they had their original data, they applied a spherical projection, and then they computed homology on that spherical projection. So up here, this is data collected from a non-blocked blood vessel, and down here we have blockages. And as you can see, two-dimensional homology, there's a lot more present down here when we have blockages than when we don't have blockages up here. Okay. So you can dive into the paper if you want, but I just wanted to present some pretty pictures on it. Also, some recognizing patterns in zebrafish. So we have some stripes here, and to do TDA, to pick out those cycles, they actually close the boundary to form a cylinder. And so this actually does form a complete cycle. And they were able to detect different patterns of zebrafish of different species. Pretty interesting application of TDA. Also, quantifying the main cycle in hurricanes. Some TDA was done to quantify what is the main cycle. So they had their pixel values, and then they did some transforms, and then they have a persistence diagram here. I showed you the persistence barcode. That was one way of plotting the data. Here we have the birth and death axis, another in the persistence diagram, another valid way to plot the data. Here we have the main cycle far away from the diagonal, which would be a long bar in the persistence diagram. And so quantifying that main cycle. So as a whole, TDA can provide some interpretable insights into the structure of data, even in higher dimensions. Like we could picture all of the data except for maybe the Klein bottle, but the methods hold perfectly well when we get into those higher dimensions. As we've seen in the Munchak example, as well as high contrast patches, we could kind of piece together the whole structure. It can also be used to analyze and classify the data, like the diversity in basketball, medical diagnosis, and biological pattern recognition, as well as segue into my second part of the talk, hippocampal replay and preplay. All right, so now going into my research. So this is a very simplified, idealistic overview of what happens in our neuroscience. So we have an animal on a track here, a one-dimensional track, and we have some sensors in the brain in the hippocampus that track some neuron activity. And as the rat moves along the track, we can see different neurons light up. And so we might observe this sequence synchronized with these movements along the track. We can observe the sequence in the rat's brain. So we can piece together what we call place fields. Place fields are in rough correspondence with the neurons that fire. And so then when we add some color to it, we can really see how the neural sequence and the movement play along side by side. So as the rat's moving, different neurons are lighting up in different areas of the track, and the correspondence of the place cells in the hippocampus and the place fields on the track. So in addition to when the rat is actually moving, when the rat is resting or sleeping, there we see accelerated sequences of past and future events going on in these same neurons. Lots of times they correspond with what happened or what's about to happen. However, there's a lot more that we don't know how to decode. Are they noise? Are they yet to be decoded sequences? Is the rat thinking about a linear track, or is the rat thinking about wandering around in a box or even a three-dimensional environment? Right? Those are all questions that we seek to answer. So that motivates this definition and this question. So this is kind of from a pure math perspective, and then we'll get into the data here in a second. So given a finite set of finite sequences, like the ones of the rat I've just shown, does there exist a set of closed balls with uniform radius in d-dimensional space, the place fields, such that there is overlap if and only if I and j appear adjacent in some sequence? So if neuron I and j are next to each other in the sequence, their place fields should be next to each other. Right? That makes sense. The rats can't teleport, so they think basically as they would move. So if they're adjacent in the sequence, we want them to overlap. If they're not adjacent in the sequence, we don't want the place fields to overlap. Right? So is there a configuration, a construction of these place fields where that happens? That motivates my definition of constructible. A collection of sequences is constructible when those sets exist, when we can get only the proper overlaps. And the goal is to find a characterization of these sequences in d dimensions. Most of my work is focused in one dimension because that's the easiest and that's what we have the most data for. So some few examples of constructible sequences. So this is a sequence in R1 that we may be given, and we can say, yes, this is what it looks like in R1. We have an overlap between, say, 3 and 7, and we have 3 and 7 appearing adjacent next to each other. These are two depictions of the same thing, by the way. This is just with bubbles and this is with segments. Exact same thing. 6 and 2 do not overlap, and we do not see 6 and 2 adjacent in the sequence. Right, so that's just an example in R1. Here we have an example in R2. So given that sequence, we would like to maybe reconstruct this place field arrangement corresponding to the rat thinking about 2, 7, 4, 7, 8, 7, 1, 5, 9, 6, 3, 1. Right, so there's an example in R2. So in order to analyze these sequences, going from this collection of sequences that's not particularly well studied, graphs are very well studied. So let's see if we can jam it into the world of graphs. And I say there is a pretty, in fact, obvious surjective function from sequences to graphs. So given a sequence, we can identify adjacencies between, say, 5 and 3. 5 and 3 are adjacent, 1 and 4 is adjacent. You can see where this is going into an adjacency matrix. Right, so 5 and 3 are adjacent. We put a 1 there. We put zeros everywhere where they're not adjacent. And adjacency matrices and graphs go hand in hand. So graphs that arise from constructible sequences in one dimension are called unit interval graphs or intersection graphs. And they're actually pretty well studied in the field of combinatorics and discrete mathematics. So you may be thinking, well, maybe everything's constructible. Maybe this is all trivial. Well, no. If we just look at R1, there's a couple simple examples, and you can kind of see how we would fail to be able to squash either of these graphs into one dimension without making things too close that shouldn't be close. If we try and squash this into one dimension, then maybe 2 would be adjacent to 4 when it's not supposed to be, or something like that. Similarly with example 2, if we try and squash this down flat, 3 would be too close to 1 when we don't want it to be close to 1 because it's not adjacent. Right? So these two simple examples show us that there are in fact unconstructible graphs, and this is a semi-interesting problem, at least for now. I hope that I've convinced you. Which motivates this general definition. So constructible I refer to in one dimension, or the smallest dimension D in which a given graph can be constructed as an intersection graph of unit spheres is called its sphericity. And this is a concept which requires proof that it's not only fun to say, but is also well defined. Maybe there is no smallest D. Maybe there's some graph out there that you can never get into D dimensions. You can never have proper overlaps. Stealing a quote from Ronald Graham, an ideal math talk should have one proof and one joke, and they should not be the same. And so now I will prove to you that sphericity is well defined. So given a graph, say we have n vertices, I'm just going to show that it's constructible in Rn. And then maybe you can get it in Rn minus 1 or Rn minus 2, whatever, but the minimum would exist if I can show that it is constructible in some dimension. So I'm just going to go for Rn and then let you figure out the rest of the minimum. So we're going to let G be a graph with V1 through Vn, an adjacency matrix A. We're going to assign each point, each vertex to a point in Rn with the coordinates of the kth row of A plus Ni. The motivation for this will become clear in just a second. So two cases that we have to show. We want to show if they're adjacent to each other, they should be close. Their distance should be close. If they're not adjacent, they should be far away. So we have the normal Euclidean norm definition shown. Now I've just pulled out the I and j terms from the sum in both cases. Just pulled out the I and j terms. And then we're going to establish some bounds. So we want the connected points to be close. So we're going to establish an upper bound. This sum, because we're talking about an adjacency matrix, this could be 1 and this could be 0. Right? So the maximum is going to be 1. And so when we square it, we still get 1. We have n minus 2 of those. Similarly, we can actually plug in the n minus 1 and 1 minus n of the corresponding rows. So we have an upper bound for if they're connected. If they're not connected, we want to establish a lower bound. And so a lower bound on a bunch of squared terms is, of course, 0. And then this is n minus 0. These are not connected, so that's a 0. And this is also 0 minus n squared is n squared. So now we just do some algebra to tidy it up. And we have our bounds looking pretty good. We can establish strict bounds and say, if they're connected, they're closer than this distance. If they're not adjacent, they're further away strictly than this distance. And I suppose I said unit spheres, so let me just rescale to complete the proof. Right, so sphericity is not only fun to say, but it's also well-defined. So these graphs are unconstructible in R1, but actually are constructible in R2. That's fairly clear. And the sphericity is 2 then, because 2 is the lowest dimension. And in fact, these two examples, as it turns out, from the discrete literature, are the only two examples of ways that you can fail to be constructible in R1. These two types, I should say. The first type, where you have a central connected to too many non-connected neighbors, and the second type, where you have a longer cycle that's not connected by a chord. Right? And those are, in fact, the only two types, which is really, really interesting. I'll detail that here in a second. But the first one will fail the kissing number test, saying that if we have a single node, there's a bound on the number of connected nodes that we can have in order to cram it into the dimension that we would like. So if we're in 2 dimensions, the kissing number is 6, because with closed spheres, we cannot arrange 6 circles around here without overlapping. In 3 dimensions, we can cram safely 12 spheres in, but we cannot get 13 spheres around a single central node. Similarly, if we have a maximal clique, we also run into the same problem if we have outliers out here. However, this doesn't generalize to R2. We can have a single central clique and we can safely fit 6 around it unconnected. And the kissing number in 2 dimensions was 6. So that doesn't generalize quite as nicely. The second obstruction to construction was a cycle here. And this graph fails the impermissible cycle test because there are 4 or more nodes that form exactly one cycle. A graph that passes that has chords is called a chordal graph. Non-trivial homology, because this is a homology talk or a topology talk, non-trivial homology in the clique complex implies it is non-chordal. Right? So this has non-trivial homology H1 and it is non-chordal. However, the converse does not hold. Example, this graph. 1, 2, 3, 4 has an impermissible cycle, but this is actually a sphere in the clique complex, and so it has trivial H1. We also have some difficulty in generalizing this topological view of the cycle test. For example, in H2, the first non-trivial homology, the first void captured is the cross polytope, the octahedron, and it can in fact be constructed in 2 dimensions. Here's the octahedron capturing the void, here's its adjacency matrix, here's a valid construction of it in 2 dimensions. So this doesn't quite generalize as nicely as we want it to, going up to 2 dimensions. And this is the theorem that I alluded to earlier. A graph is constructible in 1 dimension if and only if it is chordal and it passes the kissing number test. Right? It's a theorem I'm not going to prove now, but it's not too bad. It's just basically a lot of cases. And it is in fact possible to recognize constructible graphs in linear time, linear in the number of edges plus the number of vertices. So let's talk about some data now. So I'm going to simulate some data before I get into the mess of real data. So here we have a 1-dimensional track. The place fields are going to be modeled by bump functions. Right? So it's not, if he's in here, it's going to fire, it's a probability. And we're going to have an animal run repeatedly along a short distance as if it's thinking. And then at each point, the neuron is going to fire with an indicated probability. So here we have a bunch of sequences run. The rats run back and forth across these sequences. At each point, say at this point here, neuron 7 would have roughly a 40% chance of firing. And as it runs further and further, it would almost certainly fire. 4 and 7 would almost certainly fire here. And then as we go further on, we get the idea. The height of these bars is just for visual convenience. It means nothing as far as the sequence goes. It's just where they're at in terms of underneath the bump functions. So then we can simulate a bunch of short runs and we can get a bunch of sequences like these shown. Right, so a bunch of those neural firing sequences. Right, so a bunch of short runs along the track, a bunch of sequences generated, simulating the data that we would see. We can play the same game in 2 dimensions. Here, the place field bump functions are indicated by intensity of color, and we can generate sequences. For example, sequence A goes 8, 5, 4, 5, 5, 6, and we can go, well, it picks up an 8, then it goes 5, picks up a 4, 5, 5, 6, ends with 2s and 8s. Right, so we can play the same game in 2 dimensions. So for this simulated data, which is nice and clean and I can control, and then hopefully the real movement data, which we know the ground truth for, we know where the rat was when these were firing. And then eventually for the ripple data, which is kind of a black box, we don't know where the rat was, but we still hope to decode the sequences. So in that order, we hope to distinguish between different types of environments. 1-dimensional versus 2-dimensional, random versus walking around a wheel graph, walking around different kinds of structures, things like that. Hopefully we can distinguish between types of environments from this collection of sequence generated. Furthermore, hopefully we can recreate the place fields. So just given the sequences, hopefully we can create a realistic reconstruction of the place fields that matches up with what we see in the rat actually moving. And eventually we'll also hope to separate mixed sequences. So the neurons don't get used only once for one place. So one track could have one layout of neurons, and then those same neurons could be tied to a completely different configuration in a different track. And if a rat's going back thinking about 1 then 2 then 1 then 2, generating a bunch of sequences, hopefully we're able to separate those and recreate the place fields from them. So in order to accomplish these goals, let's go from noisy sequences to a weighted graph. We went in a pure sense from regular sequences to a regular graph. Now let's go to a weighted graph. The perhaps naive way to do it would be via total adjacency. So the weight of edge ij is just the total number of times I appears adjacent to j, then scale it or normalize it or whatever. A slightly more complex but maybe realistic connection is what I call the adjacency correlation method. It takes into account near adjacencies. So if it goes 5, 2, 6, 5 and 6 are almost adjacent. So that almost counts for the weighted graph, a little bit of weight maybe. And so it's a slightly more complicated method, but it roughly says if I see I, what's the probability that I see j in the near neighborhood? So this is on a scale of 0 to 1. So we can go from a bunch of sequences to a weighted graphs via either of these two methods. And I show that here. So from our example of generated sequences, I have a bunch of these generated, and then we can see, right? So 5 and 8 here appears to have 19 times. And if you're a fast counter, you could probably count the 19 times 5 and 8 were adjacent to each other in the sequences. Similarly here,
reminds me a bit of this reminds me a bit of the process of forming a Dinkin diagram from a Cartan matrix. I don't know why, but
okay, I'm not familiar, but it certainly is not a unique idea.
Yeah, okay.
Yeah, then the adjacency correlation method, the highest weighted edge here is 4 and 7 rather than 5 and 8. So 5 and 8 has the most edges between it, but 5 and 8 also appear a lot more. 4 and 7, it's pretty much a 100% probability that if you see a 4 or a 7, the other one will be very much nearby. Whereas we see a 5 or an 8, sometimes maybe there's a gap. Again, playing the same game in 2 dimensions, we can create our weighted graph from the sequences. And our weighted graphs are a nice thing to work with. Weighted graphs are also a great way to apply TDA. So here we have some nicely generated data on a perfect track, 0 through 11. We generate these graphs with total adjacency and adjacency correlation. I use the adjacency correlation with the TDA because it's a richer structure, right? We get those almost adjacency structures. And then scaling from low weight to high weight edges, or high weight to low weight edges, in fact, we can perform our TDA, right? We can connect those points in the same way that we connected them in the introduction to this talk. And we can form a persistence diagram from these graphs. And as expected, there's no one-dimensional homology, there's no holes. So maybe with this nicely constructible data, maybe we can say if it has no holes, TDA can tell us, yes, we're looking at a constructible graph. Here's some 2D generated data. I didn't put the sequences up there, but this is how the sequences were formed. Put it into a weighted graph, and we do get some one-dimensional homology. And if we investigated further, I suspect the homology would be 6, 9, 7, 4, this hole, and this hole. Because these are closer together, the edges will form sooner than any chord edges, and therefore we'll get some one-dimensional homology shown. So maybe TDA can be a diagnostic for us to discriminate between one-dimensional and two-dimensional generated data. Also, rats also walk around wheel graphs, right? So they have a central place, and then they walk around to these different connected edges. So we play the same game, rat walking along these edges, generating some data, giving us a graph. And we can pick out, again, some one-dimensional homology, some long-lasting one-dimensional homology, in fact, probably from this outside cycle around 0. Right, so looking good maybe for TDA. This is exciting. However, you can probably see the problem here. This tree graph, very much not one-dimensional. It's not a one-dimensional track. This would have to be either a special environment or a rat wandering around a two-dimensional environment. Right, there's no way to squash this into one dimension without getting overlaps or things too close that we don't want. So we can generate some data from a rat walking around this tree graph, and we don't pick up any holes. So this is the issue with TDA, right? Not a one-dimensional sequence, but the barcode and persistence diagram look exactly like the barcode and persistence diagram from the one-dimensional data. Right, I summarize that here. It picks up the impermissible cycles pretty well, but it fails to detect the number of kissing violations to try and discriminate between one-dimensional data and other data. Right, so then we can do some other methods. So from a weighted graph, we desire to develop a score that will tell us how likely a group of sequences is to be constructible in one dimension. So the discrete method that I use, we're going to threshold the edge weights, and then we're going to either cut or build edges based on the weight, and that has a cost associated with it, and we're going to minimize over that cost. Right? So here's an example. Right now, it is not constructible. We have this extra 2,5 edge, and so the easiest way is going to be to cut this low edge, this low weight edge, to make it constructible. And we get a normalized cost as shown, right? Indicating roughly that 5% or 6% of the overall edge weights had to be cut or built in order to make this graph constructible. Right. Just a quick observation, the complete bipartite graphs are the hardest ones to fix. You have to cut half of the edges in order to make them constructible, which is interesting. If someone wants to prove that, that would be great. Just let me know. Similarly, the continuous method for going from a weighted graph to a score. If we have a bunch of bump functions, we can look at it and say, well, if I and j are correlated 80%, maybe those bumps should overlap at a value of 80%. That's ideal. If they don't meet there, then we can pay a penalty. We can create this objective function, and we can optimize over that. Again, we end up with the same thing. Here's our same example, optimizing bump functions on the track. If we initially guess this, then through traditional optimization processes, I think I use, conjugate gradients with the trust region. We end up with this optimization. We end up with the 1, 2, 3, 4, 5. Similarly, after we would disregard that edge, we would have 1, 2, 3, 4, 5 connection on the final optimization. And that'll give us a normalized score, which would be the value of that objective function. So we have two methods that we can use. However, the continuous method is highly non-convex. That optimization function that I showed is highly non-convex. So here's a simple example, simple path graph 1 through 8. If your initial guess is bad, meaning 1, 2, 3, 4, 5, 6, 7, 8, it will settle in on a local minimum that is not the true minimum and will give us a bad score, even though it's supposed to be a good score. Right, so it's highly non-convex. However, we can use a discrete method to make a reasonable guess in order to use our continuous method. So given a weighted graph, we can cut and build edges to make it constructible as above, and then iteratively build the place field order based on betweenness centrality. Right? Definitions over here if you're curious. I'm short on time, so I won't go over it. So basically, the dark colored points have 0 between the centrality because they're not between if you were wandering around on the edges. So if we want to make an order, we would make the low between the centrality edges and then kind of build on their neighbors inductively as we go across in order to make it one-dimensional constructive. So given a set of sequences, we can get 2 weighted graphs, either through the total adjacency or correlation method, and then we can use our discrete or continuous methods to get 4 different scores. Benefits and drawbacks to each, don't have time for those in this talk. So we can generate some expected values for nicely generated data. Doing a ton of trials from nice data, we can see that these scores for all of the methods are going to be pretty low. And then we can compare that to two-dimensional data or other data, right? And in fact, I get reliable results here. And in fact, the upper bound of the one-dimensional data is lower than the lower bound of the two-dimensional data. So those are entirely disjoint for those 4 scores. So I can confidently distinguish between one-dimensional generated data and two-dimensional generated data using the 4 scores that I've shown. Recreating the place field from the sequences, that was our other goal. So given this generated data, these sequences from our graph, the continuous method gives us a nice output of what it thinks it is, right? So given these sequences, we could generate this layout, which is in fact this pretty much the same as the layout that they came from. Yay for me. Finally, sorting environments from mixed sequences. So suppose we have these 2 different environments where we have sequences generated. We dump all of them into the same pot and mix them around. My method, which I won't go into now, but is slow but reasonably good at determining and separating these 2 sequences into 2 buckets and recreating the environments in a reasonable way from where they came. That's kind of the last month of my research has been discriminating and sorting those 2 mixed environments. So it is very slow, non-polynomial time, which is a problem when we're dealing with big data. Lastly, with thanks to Sam McKenzie from the University of Mexico, who provided me with the data, we're currently trying to sort and recreate the place fields for both directions of the track. So the place fields actually form differently depending on the direction the rat is going on the track. And we're trying to sort and recreate those. Obviously, the data is tremendously messy and very much large. When I look at this as a pure mathematician, I say, this is just nonsense. However, again, this won the Nobel Prize for Neuroscience in 1971. So kind of reconfiguring my brain to say there's going to be a lot of noise. We're going to try and do our best. So there's a bunch of references, especially for the TDA application that I mentioned earlier. So thank you very much for having me.
Great. On behalf of everyone, I would like to thank Jerome for the talk. Are there any questions?
I have kind of one, I don't know, it's maybe a little tangential. Well, first of all, thank you for your wonderful talk. That was awesome. I was kind of wondering about what is this sort of multi-parameter TDA? So I kind of understand the varying the R parameter, but what would another parameter look like?
Yeah, certainly. So I've introduced TDA in a way I was introduced to it, which makes sense to me, which is in a very geometric way with, as you say, growing that radius, connecting the shapes. I think that's very intuitive and a great way to see it. But lots of times, such as with the weighted graph, there's not any necessarily inherent geometry in the graph or a weighted graph. We can just connect it via the weights of the edges. There's other situations where there's multiple things that vary. Say if in both, say if you're scanning from bottom to top over the data or scanning from left to right and finding some way of connecting those simplicial complexes, or instead of connecting a chain complex in a straight line, suppose you have 2 parameters varying on 2 axes and making kind of a grid of chain complexes. I don't have an example that comes to mind right away for a physical interpretation of 2 things varying at once, but that's the idea and that's how it's constructed, yeah.
Perfect, thank you.
I was wondering, also thank you for the talk. I was wondering if you knew of any connections between the methods that TDA uses and like some ideas behind like neural networks, because it seems like both of them are in some sense embedding high dimensional manifolds into like a lower dimensional space for analysis. Do you know of any connections there?
Yeah, so there's a ton of things that can kind of go back and forth between TDA and neural networks. As I referenced when I was talking about the diagrams and things, you can plug the data from the persistence diagrams right into a neural network and train it to discriminate between types of data. And then you can take the output even from a neural network and do TDA on it and kind of go back and forth to your heart's content. There's some very interesting data visualization, and as you say, compressing high dimensional data into low dimensions. We all know, hopefully, about persistent component analysis, which is a big deal if you're trying to preserve the global structure of the data from high dimensions. PCA will do what's best for that. However, if we're trying to preserve the actual topology of the data, I went to a recent talk, I forget who gave it, about having data points sampled from a mammoth. And when you sample that and do persistent component analysis on it, when you compress it into 2 dimensions, it looks nothing like a mammoth. But there's programs now, UMapper, or UMap is one of them, that desires to take that high dimensional data and compress it in a way that preserves the inherent topology, the holes and the things like that in the data. And that's some really cool stuff going on there. So UMapper is one thing that I would recommend you check out if you're interested in that.
Very cool. Thanks.
Mm-hmm. Let's see, one in the talk. Let's see, from Daniel, very good talk. Thank you. I have a non-math question. How did you measure the NBA data? Can you elaborate on that? Did you study clustering or were there higher dimensional homology? Great question, Daniel. I would love to talk more about basketball. So as we scroll through my numerous slides here. So my main point of that, it was for AATRN, was the YouTube initiative. And so you can find it there and watch the whole video if you like. But my main point, as I was saying this, is I wanted to study the diversity of playing styles. Playing styles as measured by 3-point attempt rate, rebounding, number of steals, number of blocks, things, usage percentage, how often the player shoots, not necessarily how well they shoot, but how often they shoot, things that indicate playing styles. Because my conjecture and observation, and this is supported in many types of data, is that there was more diverse play and more diverse players back in the 70s and 80s than now. Because right now, it's a huge emphasis on efficiency. And so players have become more homogenous. Almost everybody shoots 3-pointers now. There's no like a Dikembe Mutombo who specializes in blocking shots, or say Larry Bird that shoots 3-pointers at 3 times the rate of the yearly average. And so, as I said, as my analysis indicates, through the average persistence of age 0, when we go throughout the years, older teams, more diverse, more spread out when it comes to playing style as measured by the characteristics. And there's 7 of them that I take a look at the video: rebounding, 3-point attempt rate, steals, blocks, usage percentage, and others. So it's a 7-dimensional point cloud that I run the big analysis on. There's also a quick allude to the 1-dimensional homology. So if we look at the 2016 Warriors here, and I make a bigger deal of this in the video, we could see that if we compute 1-dimensional homology, there's going to be a hole between these 4 players. And it's a little bit cheesy, but I wave my hands and say they could fill the hole in their roster by putting someone about right here. And if you know your NBA history, the 2016 Golden State Warriors, one of the best regular season records. However, they lost in the championship. The 2017 Warriors, the next year's Warriors, they added a superstar named Kevin Durant. And in fact, he fit in exactly right there to fill in that hole in 1-dimensional homology, which is maybe a coincidence, maybe a stretch, but I have fun with that in the video.
How did you compress the 7-dimensional point cloud into 2 dimensions?
So what's shown here is only 2 dimensions. It's 3-point attempt rate and rebounding. And so on these axes, so it's the rebounding, so this blue point here, so Bill Walton, he rebounded at a rate twice the yearly average. Okay. Larry Bird shot 3s at a rate 3 times the yearly average. As I talked about in the beginning of the talk, TDA is great because we don't need to visualize the data in order to do analysis. The Viator's Rips complex is defined completely from the pairwise distances. which are easy to compute in 7, 8, 10, a million dimensions. Super easy to compute distances. So it's only for visualization purposes that this is in 2 dimensions. The 7-dimensional analysis runs perfectly well, and you can talk about average persistence and H1 and things like that and do analysis on it without being able to visualize it at all.
Mhm. Are there like more patterns that are there in the 7 dimensions than there are in like the 2, and just looking at these 2 parameters?
I mean, yeah, the data set is certainly more rich. You can have higher dimensional homology. Obviously, you can't. You can't have 3-dimensional homology in 1-dimensional space, and so there is, in a sense, more to go through. However, if the sampling wasn't from necessarily a manifold where there's a big hole or something like that, it's hard to detect. So there could be a lot of analysis there open, but my work is focusing on 1 and 2 dimensions, 1 2-dimensional homology, I should say, with the 7-dimensional data cloud.
Cool.
So you mentioned in your other methods towards the end that the current method that you have for sorting between the sequences is fairly slow. Do you have hopes for improving on that?
Maybe is the short answer. It's going to be tough because I don't know if I'll ever be able to get it to a place where it's entirely applicable on millions of sequences. For this data set, to create this one, it ran in 2 minutes, which is great if you have 7 nodes and 20 sequences. But if you have 50 nodes and a million sequences, that doesn't scale particularly nicely at all. And the tough thing with sorting these is I think there's 24 or whatever to start with. And my naive method when I started this goal was, well, there's not that many of them. I'm just going to try every possible way of putting them into 2 different buckets and see which scores best, which is roughly 2 to the 23rd power, which not great combinatorially. And so what I've done in order to speed it up a little bit is I've looked at the graph that all of the sequences create. So big mushed up wouldn't necessarily make any sense on its own. And I said, what's the most weighted edges? And of the most weighted edges, are there a handful, maybe 4, 5, 6 sequences that capture the most important edges? And if I can get that down to 6 or 5 or whatever, then it becomes feasible to try and sort out those 5 or 6 by exhaustion. And then from that, I have this big line of sequences, and I can say, I have these environments from the sorted out small bucket of sequences. I can say, do you fit better in bucket A or bucket B? Okay, go in bucket B. Do you fit better in bucket A or bucket B? Okay, not either, go at the end of the line. And then I can keep sorting the rest of the sequences based on those few that I selected. But as I said, very much non-polynomial. Hopefully, maybe we can get it synchronized or something like that to try and make it actually applicable on the real data, but we'll see.
Yeah, definitely cool to hear where that's at. Are there any other questions?
Yeah, so probabilistic Bayesian split for those buckets. I've spoken to some probability theorists on this, trying to get some kind of improvement. But the method that's the aspect that's a bit unique to this is as they're being sorted, they're actually building the ground truth stronger and stronger. So if there's only 3 or 4 sequences in each bucket, and you throw in a new sequence, that new sequence accounts for whatever it is, 10% of the overall data. And so it can alter the formation of those place fields pretty heavily. But if you've sorted a million sequences, one new sequence doesn't make a difference. And so kind of building the ground truth as they're sorted is something that is an aspect that is not playing nicely with the probability theorists.
All right, I'd like to thank Jerome again